And he has significant work in developing strategies for synthesizing biomolecules, different types of biomolecules against different diseases, including anti-cancer. And I, as a Worst Fellow, in collaboration. Hello, those people who are joining online, please mute yourself, otherwise there is creating some noise. Okay, so as a Bose Fellow, I feel proud to share with the recent development of Professor Misra's research. They, in collaboration with their divisional colleague, they have developed a new strategy to target the anti-cancer molecule exclusively to the breast cancer cell. So this is an outstanding research area, and now it is under a translational process. So I congratulate Professor Misra for your outstanding achievement. Thank you. And now he will address his welcome to the gathering, please.
So good afternoon, everyone. I think I shouldn't desert this place to welcome you, and all of you know probably that our director is out of station. So, and also please pardon me because of my cat voice. And our director nominated someone to address you, and he also has another engagement. So ultimately, I was asked to do this job, but I feel pleasure to be here to welcome you to Bose Institute for this important lecture. And it's one of the important lecture series of WAST and West Bengal Academy of Science and Technology. And although, as Didi said, there are many Dada Didis around, I can see, we generally call in Bengali, although you are a Bengali but situated in Pune, so we call Dada and Didis. So I, since I am not an expert in this area, so it's only my part is to welcome you all, and you all know that Bose Institute is has a strong legacy to welcome or to foster the activities related to science and technology and research development. There are many people who are sitting in this audience. Many of you are already fellow of this academy, and many of the who are sitting far from this place are going to be the fellow in future days. And I expect that everybody will be enriched with your lecture with the proposed topic. Although I know only the potato is one important part, and I personally work on carbohydrates, also not in the energy storage molecule, but for some other purpose. At least I know that potato has starch and we need it every day. So that is important thing. So with this, I request the President and the Vice President of West Bengal Academy of Science and Technology, together with our Didi, the Secretary, and Sampati. Of course, our Arjun Bose Chanjini, always the evergreen Orun Laidi Majumdar. So I expect that you will be benefit with your talk, and I request all of you to attend or someone who are coming to attend the lecture and enjoy the event. Thank you.
Thank you, Professor Misra, for your hearty welcome. Thanks to the director also for giving us the opportunity to organize such a prestigious lecture here in this hall. So thank you so much. And now I request Professor Dr. Amit Ghosh, the Vice President of WAST, to welcome the gathering. And you all know Amita does not need any introduction. But still, for the students' sake, I tell you that Amita is a very renowned scientist and he is vice president and he is holding different positions in number of academies. So maybe he is in the, you can say that he is in the policy decision making position, and he retired from IMTech Chandigarh. He was the director of IMTech Chandigarh, and after retirement, he joined Gandhinagar Institute for DDT also as advisor and scientist. And presently, he is emeritus scientist in NICET, the Cholera Institute. So he had some old connection with Bose Institute also. At the early career, in his early career, he used to work at Bose Institute as postdoctoral fellow in biochemistry department. So he had some connection with Bose Institute also. So now I request Amita to address the gathering on behalf of West. Please, Amita, please.
Well, actually, I'm very pleased to welcome you all to this endowment lecture. I'm really sorry because of some personal problem. I cannot be there in person. And I wish our president were here, but due to exigencies of work, he's also had to go to Delhi. Now, although the most people who are present here know something about our academy, but still I thought for the benefit of students and the others who may not be very familiar, I may spend a few minutes to talk about your academy, which normally is done by our president. Now, this academy was founded long back in 1989, and the first chairperson was Professor Shushil Mukherjee, and the vice president was Professor Shobhtesh Pakrashi. Although it was formally founded in 89, talk about forming this academy to spread awareness of science among people and for the welfare of our state was going on for quite some time. In fact, it started in 86, if I'm not mistaken. Now, what does this academy do? Well, if you look at its objective in the website, the 6 or 7 are listed, but if I formulate them and just them in one sentence, it's primarily involved in spreading awareness of science among students and the society at large, and also for the welfare of our state. And this it does by having, as you have one today, by endowment lectures, which are given by very prominent people, annual orations. Like this last year's annual oration was given by Tessie Thomas. You may know she was she headed the ISRO's, electrical propulsion system or something like that. And thanks to our president, he could convince Dr. Thomas to come over, just like we have today a speaker from outside Kolkata. So this academy spares no efforts to actually give meat to these particular functions. Now, we had this particular endowment lecture about Professor Sengupta last year. It was done by Professor Paramjit Khurana, again from Delhi University, outside Kolkata. So all I'm trying to emphasize the fact that although we are West Bengal Academy, but our reach is quite large, and we do not fight shy of inviting prominent people who have done remarkable work in their own fields. Apart from this endowment lectures and this annual oration, it also takes part in a lot of outreach activities. Now, it's an unfortunate fact that today students are losing interest in science. Many places, we also, in connection with another organization, go to different parts of West Bengal and find that in some places they have actually folded down the science departments because there are no takers. And this is unfortunate because if you come to think of it, this state produced a number of very, very prominent scientists, starting with Jagadish Bose. Even in the Mahendra Lal Sarkar established cultivation of science in the ways of Royal Society. And in that same state, if people are not interested in physics, chemistry, biology, mathematics, it is unfortunate. But does it mean that why they're losing interest? Now, academia has found that if the students are talked about science in an entertaining way, by entertaining, I don't mean it's diluting its contents, then students are interested. For example, in 2024, this academy went to different places. You know, it went to Durgapur, it went to Uttarpada, it went to Jhargram. And judging by the, I wish some slides could be shown, interest of the students, the question answer sessions and the way they sort of mobbed the speakers, it was very, very clear. The science, if it is given in a proper form, the students do not lose interest. They lose interest because it is given in the form of notebooks, in the coaching classes where the content of science is lost and the end point is marks. Maybe there's some societal reason for that, but whatever be it, the point that one tries to make here is that all of us have a duty to see that the noble tradition the state had in the area of science should not be lost. And these are just not the lectures and they also run workshops. For example, because of this personal connection with the industry and other places, Professor Chakraborty could arrange a training program on Internet of Things. And you'll be surprised to know that it's generated such great interest that people from faraway places like Jalpaiguri and other places, they also came and joined. The unfortunate part was that, and it was done in collaboration with Jadavpur University. Of course, they are the main players, their department, and a company called Exonics, who are expertise in this area. And the enthusiasm was so much that President had to give an assurance that there'll be a repeat of this. Whether this is possible or not, I don't know. But just to say that 136 participants applied, and many of them had to turn back, not because they're not good or anything, just because the logistics becomes a problem. So all I'm trying to say this is, this academy in its part is trying its best, just not to spread the awareness of science, getting involvement with the commercial enterprises, which was not there before, and making quite a success of it. Now, I can of course go on and on because I have records of the lectures that has been given. I can name them and go on and on, but there's no point because I always remember what John Kenneth Galbraith, you may have heard about it. He was a professor of economics, very prominent economics professor in Harvard. He was a ambassador to India. He told President Kennedy that he knew of no such people other than Indians who love to talk more. So I always remember that, and therefore I try to keep things brief because people are not really interested in learning a catalog of what we have been doing, but rather in knowing in gist what the academy is all about. But before I finish, I just like to make two very brief comments. First, any institution grows or dies depending upon the interest of the people who are in the institute. Infusion of young blood or disinterest. I gave you the example of science stream. Students are losing interest. Therefore, many prominent colleges are actually finding it difficult to fill up their seats. Similarly, our academy will thrive if newer bloods come in because all of us one day will pass away. And for this, an active participation of the existing fellows is very, very important. Not only just by the way of attending these lectures, but also by nominating people. You'll be surprised to hear, I will not name the person, a very, very prominent and a physics professor is not a fellow of our academy. It's such a, strange thing for me to know this. But the reason is people are reluctant. Once we get elected, somehow, I don't want to use any strong word, but do not like to exert our effort. So request is please nominate people. By that, I don't mean that anybody who gets nominated gets elected because we have 13 sectional committees which go through a very rigorous method of selection. And then people become inducted as fellows. And this is the way the academy gets enriched. And in times to come, it will flourish more. But before I finally finish, I'd just like to say a word of appreciation for our current president. Our academy has always been very blessed with very prominent, distinguished people as presidents. But after Dr. Chakrabarti took over, this has gotten a new dimension because, as I mentioned, the connection with industry, actually inducing industry to contribute funds, all these have happened during the tenancy of the present president. Also, now you can see all these things in the website. We have a web link. All the lectures are uploaded. So in case somebody is interested, one can go. So in other words, what I'm saying is it is going from strength to strength. And if it has to go stronger, then as I mentioned, more active participation of the fellows in the way, if for nothing else, in the way of nominating promising people, giving encouragement to young scientists to come and join. And then they can, of course, personally take part in these activities and make our already great organization greater. I'll stop here and I will request the Professor Dr. Shampa, I mean, I'll call her Shampa, so very much.
Thank you, Professor Ghose. Thank you for your very nice introduction about WAST. Thank you so much. So today Dr. Ghosh also told that today we gathered here to hear the J.C. Sengupta Endowment lecture. But many of the students may not be knowing who is J.C. Sengupta and why there is endowment lecture, why he is so famous. So I request Professor Kumar Palit to say briefly about Professor J.C. Sengupta, who he was and what he did, and why we have arranged WAST is arranging this kind of endowment lecture. Professor Kumar Palit is an ARS scientist of CRISA, the Central Research Institute of Jute and Allied Fibers. He retired from there. And at the last stage of Professor J.C. Sengupta, he also worked with him. And later on, he also continued some work of Professor J.C. Sengupta. So he will highlight and why this endowment lecture is organized by us and what is the history of that will he explain. Professor Palit is not only a scientist, but is a very important social worker. He runs a very remote, in a very remote village of Sundarban. He is running some free school for primary section for the children, and also the mother and elder sisters of the children. He is running a school for the adult women, and not only giving them academic knowledge and academic credentials, but also for cultural activities. He also nurturing in them the cultural activities, different songs and different dance drama, and so they are culturally also enriching with the help of Professor Palit and his very important and reliable wife, effective wife, Dr. Dhira Palit. They both run these facilities at remote part of the Sundarban. So now I request Professor Palit to highlight about Professor J.C. Sengupta.
Thank you, Professor Bose. It's a great pleasure for me to say a few words about a great plant physiologist, educationist, and an administrator of our time, Professor J.C. Sengupta. Many of you must not be knowing about him, such a great person. Now I'll read it out. I have jotted these points for this meeting. You will excuse me if I read it from my note. Professor Jyotish Chandra Sengupta was born on December 18, 1900 in outside. That means this will be his 125th birth anniversary. The outside is a village in Bikrampur of Dhaka district, now in Bangladesh. His father was Jyotish Chandra Sengupta, was an engineer employed in the railway service. His mother, Srimati Surama Sengupta, was a prominent pioneer in women's education in Bengal. On graduating from Bangabasi College, Kolkata, in 1921 with botany honors, he joined the postgraduate course in botany at the University College of Science, Baliganj, Calcutta, where Paul Johannes Brühl, a versatile German scientist, was developing the department as the pioneer center of study and research in plant science. Brühl arranged to send Jyotish Chandra to Heidelberg in Germany to work with Professor Ludwig Bose. While in Heidelberg, Jyotish Chandra worked on rheotropism, tropic movement. Rheotropism, the mechanism of bending of roots towards the current of water. He established that such movement of roots was not due to the hydrostatic pressure, but in response to certain electrolytes. He also observed that the accumulation and localization of different ions and electrolytes in specific cells help the plants to adapt themselves under different environmental conditions. This is surprising. These were really fascinating observations almost 100 years ago, as it is only recently that we have realized the importance of ionomics involving elemental compositions of a plant to address biological problems. Jyotish Chandra returned to India in 1929 with a PhD in natural science from the University of Heidelberg and joined the Department of Botany, Presidency College, Calcutta, and started researching on the physiology of plants from diverse ecological conditions, like the salt-loving mangroves of Sundarbans, plants adapted to high altitude of eastern Himalayas, and other plants of Bengal plains. These studies made him the first ecological plant physiologist of India. In 1930, Dr. Jyotish Sengupta married Nilima Gupta, a student of Loreto Convent, Calcutta, and an accomplished singer. He became principal of the Presidency College in 1950. In 1952, he became the Fellow of National Institute of India, which is now INSA, Indian Institute of Science. Indian. He was elected President of the Botany Section, Indian Science Congress, at the Baroda session in 1955. Jute at that time was one of the foremost foreign exchange earner crops, earning crops of our country. Jute is grown, many of you may be knowing or don't know, I don't know, because jute is not researched very much. In Bengal, many people avoid jute research. I don't know why. But jute is grown in the eastern parts of India during rainy season of the summer months, from late March to early September. Jute growers at that time faced a serious problem in that whenever they sowed jute seeds in March, the plant started flowering within a month. Since jute fiber comes from the stem of the plant, the stunted stem growth due to the onset of early flowering and branching hampered the yield as well as the quality of jute fiber they produce. No agronomic intervention could alleviate this problem. Professor J.C. Sengupta, through his meticulous experimentation, demonstrated that for flowering, jute requires a critical dark period of 11.5 hours. This is exactly what is available in this part of our country during March. He recommended sowing to be shifted from March to early after 15th of April. Thereby, all the problems of early flowering were solved. This was a remarkable observation that saved our country and the jute cultivation in West Bengal. His conclusion that jute requires a short day photo period for flowering, but a long day conditions for the growth of the stem was adopted by the agronomists and plant breeders to change cultural practices and the developing high-yielding jute varieties. Considering Professor Sengupta's love and contribution to anything and everything related to plants and extensive administrative experience, he was entrusted with the revival of the Botanical Survey of India, BSI, as its chief botanist in 1955. He, along with Dr. M.S. J.K.E.K. Janaki Ammal, another renowned botanist, thoroughly restructured BSI by opening several zonal stations all over India. He established a liaison with the Kew Botanic Garden, London, as well as many Indian organizations like IISER, CSIR, ICMR, etc. Thank you. For better functioning of BSI, M.S. Janaki Ammal and he were instrumental in initiating the publication of the updated account of Flora of India, Independent India. Thank you. The then government of West Bengal entrusted Jyotish Chandra to organize the state's higher secondary education policy as the administrator during 1963 to 1967. He developed a curriculum which is regarded as one of the best preparatory steps for undergraduate and postgraduate studies in a variety of disciplines. I'll deviate a little bit. He is the person who emphasized on taking physics as a subsidiary subject with botany and chemistry. Physics is a must that time, or in the zoology, physics and chemistry, whatever may be. Not they're both botany and zoology, botany and both are biological science. No, he emphasized on physics. Only one student I remember, that is Professor S.P. Sen, Chamapada Sen, was allowed in Presidency College to take botany with physics and chemistry. Maybe, sorry, I'm so sorry. He also took anything. Professor J.C. Sengupta was closely associated with early organization and setting up of the Birla Industrial and Technological Museum in Calcutta. He also took active interest in the activities of Asiatic Society. His vast experience and farsightedness benefited practically all major institutions in the post-independent India. He came back to his alma mater, the University College of Science and Technology, Kolkata, in 1968, as a retired scientist of CSIR, to continue his research on photoperiodism, the subject he was so fond of. On January 21st, 1969, he heaved his last on the feet of the alma mater. Paliganj Science College, Shirit Tolai, Marajan. His wife, late Nilima Sengupta, took great care in donating Professor Sengupta's large personal collection of rare books, journals, periodicals, and valuable reprints to his department, to the Department of Botany, University of Calcutta. Today's endowment lecture is also through her benevolent contribution. I am personally indebted to late Professor Yashodhara Bagchi, his daughter, and late Professor Amiyo Kumar Bagchi, his son-in-law, for sharing many personal anecdotes and for providing many of his publications by the late Professor J.C. Sengupta. Thank you very much.
Thank you, Professor Pali, for enriching us, all of us, to the history of Professor J.C. Sengupta's endowment lecture. Now, the time has come for the actual endowment lecture, will be delivered by none but Professor Anjan Kumar Banerjee from IISER Pune. And we are very lucky that we got him as the orator of this endowment lecture. Now I introduce Professor Moitreyi Dasgupta, famous, the Moitreyi of all of yours, Moitreyi, the Secretary of WAST. I request her to, you all know her, so I don't need to introduce her. Professor Moitreyi Dasgupta, please introduce today's speaker. Thank you.
Thank you, Shampadi. I'm very happy on behalf of WAST. I need to express this happiness to see so many of you around. It's an exceptional sight, and I'm very happy that Convener Professor Shampadi has taken this decision to have this endowment lecture held in Bose Institute. I'm really happy to see this. On behalf of Academy, also, since I'm the secretary, I need to extend the thanks to all of you who are present here. And now I come to introducing Anjan, Professor Anjan Banerjee. And first I'll read out what he said, and then I'll talk 1 minute about his science. Okay, first, because this is impossible to remember. And he also didn't mention everything. I couldn't match his CV with what he has written. It's very, very few things that he has written here. So, let's see. Can you light me? I also need to, I did express thanks to Bose Institute. Professor Mishra, I did express thanks to Bose Institute and my personal regards to you. Thank you. So Professor Banerjee is currently a faculty member in the Department of Biology at IISER Pune. He carried out his research at NCL Pune, obtained PhD degree from University of Pune. He had a stint of 7 years of postdoc in Iowa University and worked on fundamental mechanisms of potato development. So potato, apart from starch, has something to do. As an independent faculty, he joined IISER in 2008. He is an elected fellow of all 3 prestigious academies in India, recipient of Tata Innovation Fellow Awardee, elected member of Indian GRC. He is a life member of Indian Plant Cell Tissue Culture Association and endowed with institutes Rahul Bajaj Chair Professorship. There is no end to this. Okay, so I'm not completing this paragraph also. I'll come to science. And his primary research interest is long distance macromolecular signaling and plant developmental biology. And he uses food crops and extant land plant cell models and blah, blah, blah. Let's stop here and I'll talk about what he does. So I know Anjan for a long time, and we have been participating in different meetings in different capacities everywhere. And it's like decades. And it's like decades that I actually know him. So what he does is, he does plant developmental biology. Now, we know that roots go down and shoots go up. This much, even if you are not from any plant background, you know. Now, these are different developmental programs, right? To make shoot a shoot and root a root, right? Now, what he does is he takes a example of potato, which is like a stem, but it grows underground. Any exception in biology is an opportunity, right? Because then you have a bunch of systems, right? So he uses this to explore how much of how is it possible that a shoot is growing underground, and what type of programs are common between them. So he uses that. That's one. Number two is he discovered those plants and programs that involves microRNAs that communicates with shoot and root and actually helps in development of this potato. So apart from starch, there are many other things in potato development. Potato is underground, but it's shoot. This much you understand. And finally, what he does is whatever he discovers in potato and all, he uses Viscometrilla patens, which is a very lower plant. Okay, so what we do, any developmental biologists, we go down and down through evolution to the basal models to see how much of it is a core program. There are ornamental developments as you grow up. So when you're properly developed, you have so many other things that are developed. But when you have the core program in the root and in the basal plant, then you know how it evolved, how these cores were arranged. So he uses Viscometrilla patens to again, establish what are core programs. So this is what Anjan Banerjee does, and I welcome and thank you so much. It's a pleasure to welcome you here from my personal behalf.
Share option to the allow करते बोलो allow भी कैसे देखो sure
So, good afternoon, everyone. I'm really very fortunate and privileged today to have been invited by WEST, the West Bengal Academy of Science and Technology. I'm really grateful to all of you, particularly Sampati, Maitri, those of you have asked me to come here. It gives me a great opportunity to share our views, our opinions, and our perspective in understanding a different field of science that we have been doing it for the last 25 years, including my 7 years of postdoc. So I'm really grateful to WEST. And today I come here in a time when you are 125th anniversary of Professor J.C. Sengupta. It's really a great opportunity for me. I was listening very carefully and patiently when Professor Mr. was sharing his observations, particularly in jute. What was that? And I was trying to thinking about how it correlates the science that we do. And I'll give you glimpses of examples. So today, what I decided when I thought about giving a talk here, yesterday I gave a talk in IISER Kolkata, mostly in UG students. I didn't change my slides, I thought. And I was told that it will be a kind of a heterogeneous crowd. So do not make it very, very research, data intensive, rather make it a more of a concept oriented. The letter that Professor Chakravarti, whom I met during the INSA inductions in Chennai, SRM, and we had a brief chat together. And so I'm really privileged, honored. Thank you all for giving me this opportunity. The title of my talk is Impact of Mobile RNAs and Proteins in Plant Growth and Productivity. What I have decided today to give you an overview of the field of the last 30 years, including my 25 years of work in the same field. Now, when I say 30 years, you can think about what he's talking when you talk about the mobile RNAs and proteins, right? This concept itself, the mobile RNAs, mobile proteins, mobile signals, was certainly something was not known to the physiologists in the community of Bengal. And how it evolved, how it came up, and that is what I'll give you. So first, roughly about 20 minutes or so, you will give. I have very carefully picked up the best examples and the best papers in the science today to let you know that how the science originated in this field. And this is not something very old. This is 30 years old. I'll give you the example, and you will appreciate that what people have thought about how the fundamental hypothesis led to a different sort of perspective of understanding plant physiology, in particular in the context of light, in the context of temperature, in the context of biotic stress that you can think about. So that's the title. Let's start. Please feel free to stop me any time. I understand many of you may not be from plant science, but those of you who understand, wonderful. If not, raise your hand. I will be happy to answer your questions. Do not hesitate to ask me. Okay, so let's move on. Thank you. So this is what it is. Of course, as Maitri introduced, I work on this model plant. I've been working for, as I mentioned, 25 years. So let's see what is our passion, what are our questions in this plant, and what we do and why we do. Does it really make sense for us to do this? Let's continue the investigations on these plants. We have, over the years, we have closely, roughly about half a dozen mobile signals we have discovered from this plant. Question arises in our mind that why this particular crop requires that many mobile signals? I mean, in what context? Does it really make sense, or these are just something by diffusion that we are just identifying? Absolutely not. And I will give you examples of findings or crucial findings that carefully establishes in the field that this plant, particularly the potato, as I said, is a classic example. It requires mobile signals. Not only this plant, we have all the plants today in the field that we know. These signals, by the way, before I start, these signals are not omnipresent in the plant. These signals are the signals carefully generated in the plant in the integration of environmental factors and the endogenous factors. Plants' intrinsic signal and the exogenous signals together integrate, and plant generate carefully the mobile signals. And I'll give you the glimpses of examples from many other pioneering works today. I'll let you know that what people have done and how it has established the concept. Plants are sessile organisms, all of us know. Unlike animals, they cannot move, so they need to live throughout their life in the same place, which allows plants, which gives plants a different sort of challenge to to sustain in a place where if the nutrient is deprived, water is deprived, high temperature, an animal can run away. Just imagine, temperature rises, animal will run, the plants cannot. Now it does mean that it has no, that's the plant. Now this plant requires different sort of signaling system. That is what I'm trying to point out here. It is not something a very by default system. The plants require spatiotemporal signals to deal with different kind of situations, whether there is any kind of stress. Now, so this is human physiology for students to understand. Did micro injection to the mesophyll cell. Micro injection of mesophyll cell, within few minutes, the knotted one moved all over. This is the one, and this is the mutated knotted one. So there is a control. It does not move. Wonderful evidence. That means you have a protein which moves right away to your peripheral cells. On the same side, on the right hand side, you have the mRNA movement. No, not it. It's a protein movement. It's a knotted one mRNA movement. In the same way, they showed that these two evidence, first evidence in the community, the transcript moves from one cell to another, because they are required to reach to the next level of cell layer in order to establish the gradient, in order to control the multiple other factors to maintain the stem cell maintenance in the sugar. You can follow, right? That's what I'm trying to indicate here. This is the first evidence, by the way. Okay, so now move on. These are controlled. Okay, these are all controlled. The antisense and the antisense one, you don't see the signal here. You see the signal here. You see signal in both cases. These two are controlled. Here also these two are controlled, and these two get the signal. So protein and RNA both move. This is the summary. KN1 mediate the selective plasmodesmodal trafficking of KN1 cell. Protein is moving its own mRNA. Wonderful discovery in the community. The same protein is moving its own mRNA. Okay. The first endogenous transcript is reported to move cell to cell systemically in the plant. Developmental outcome: it controls the stem cell maintenance. We reviewed. I took a chance. I said, let us review the 25 years from 1995. And I asked my student, I said, let us review. And we reviewed from our lab, because I've been working on the field. If you see here, you have the SAM maintenance, you have the phloem transport, you have the leaf development, you have root growth, tuber formation, seed development, germination, biotic stress, flowering.
You tell me any phenotypic traits, there are examples of beautiful examples of mobile signals which are contributing to that trait. You mutate that signal, the trait is disrupted, trait is affected. Clear evidence in the community today. So we reviewed this. I encourage students to look at that, all the signals. By the way, these symbols on the right and left, it denotes the protein signal, the mRNA signal, the siRNA signal, the mRNA signal, the microRNA signal, the LNCRNA signal. After 30 years, amazing gene regulatory mechanism. LNCRNA's gene regulations was not very clear to us. Today we see LNCRNA is critically making the gene regulations of the developmental outcome. Now, LNCRNAs are also mobile signals. Not all. Not all. There are very few selective signals are now known. Let's move on. So, what has contributed to this field is that, if anything has contributed to this field, is grafting. The grafting, phenomenally, traditionally, agriculturalists, all the people in the breeding system, they have been doing the grafting. You see the paper which was published in last year, Molecular Plant, is a very high-ranking paper. You can imagine what the group has been highlighting here. You have plethora of grafting techniques. These grafting techniques people use decades for improving the crop traits, be it yield, be it productivity, be it growth, be it vigor, be it root, whatever you consider, doesn't matter. People use grafting, cross grafting, this grafting, that grafting. This grafting has set the foundation today for plant biologists to understand that what is happening, what is the signal exchange between the two. So we should understand. And the mechanism that we now, if you look at the paper, you will see that how the grafting is allowing the developmental signal exchange between the stalk and the sign. Amazing, really. Look at this example. You have two examples here. One is a stem grafting on the top, and you have a green one, which is a source, and this is sink, and this is your source. This is basically expressing a mobile signal, and you see after the grafting, what happened? The signal has moved from the source to the sink, and as a result, your leaf has turned completely red. Okay. This is a clear example. It has happened through the grafting. Now this signal has movement with a purpose. Please remember this fact. There are beautiful papers. I encourage you to look at it. They move with a purpose, and those signals were not there. They got generated, they got synthesized, and they got moved from place to place and affected the development. Similarly, here you see there is a limited mobility, there is a high mobility. What it indicates, if you develop plants with a dominant negative, I hope you know what dominant negative is, which controls its own genes and repress it. Thereby you see that the signal is moving from the root. It's a root grafting, by the way. It is grafted here. It's a root grafting Arabidopsis, and that signal from the root has gone the entire plant for a reason. Okay, movement not without a purpose, movement with a purpose. Okay. So we'll move on. And another thing I must mention here, what matters to understand for us, for biologists to understand this kind of signaling system is quite challenging, because it's not that I go tomorrow and make a graft and you will say, okay, he told me and I am unable to find the signal. That means either I am wrong or you are wrong. Because there are critical factors. Which are those critical factors? I have written on the right hand side. Time of grafting, strength of signal, developmental stage, limited or maximum mobility. A plant developmentally completely old and you have gone and made a graft, it is unlikely to function. A plant is too young, you have made a graft, unlikely to function. You have to see the signal potential, what the outcome you're looking for. All these factors on the right hand side, this is a fantastic work from Kregler in Germany. He has done that, and they have written a Transient Plant Science Review, whereby they showed that timely grafting matters to catch the signal. It is in the biotic stress or it is in the abiotic stress. You can really find out that what is happening. The field has expanded like anything. Okay, since I said in the beginning I will share others' work, then only I will come to my work. Okay, I think it's an opportunity for all of us to see and appreciate the work. Tracking the messengers, how to track the messengers, big challenge for biologists. Okay, these RNA molecules move, these proteins move, but you can say, okay, you can track it. Understanding short distance signal movement is a challenge for us. Okay, you cannot simply do something and find out what the signal is. It is going to get a lot of noise. You won't be able to distinguish which is the true signal. So on the right hand side, you see a number of techniques have tools, particularly some of these are very recent edition, 23, 24 edition. All I knew earlier, when I started working, all I knew that there are polypeptidine tract binding sequence in the RNA loaded with polyUs, and thereby the protein binds, makes it a complex, and then this complex go from one cell to another, and finally to the target cell. But lately, all these special mechanism, you see TLS sequence, tRNA-like sequence, tRNA-like sequence, polypeptidine tract binding sequence, cyclophilins in Arabidopsis, vesicle mediated signaling, 5' methylated cytosine, ribosomal RNA processing proteins, and exosome mediated. All these mechanisms, now people have discovered that how these signals go from one source to the other. Okay, right? It's a, if you see the inventions and the findings are really very fascinating. One more thing I want you to know, particularly the students, imagine cell will make thousands of mRNA, right? So are thousands of mRNA signals? Not necessary. There could be only one signal. There could be only two signals, right? Now the point is, how would plant identify that's the signal, right? Can you imagine the challenge for us as well as for the plant? Not only that, the direction of transport is completely unknown to us. Which way it will go? Not known to us. Now, the recent discoveries clearly highlighted the science discoveries. I'll show you the science discovery. They have identified cyclophilins in Arabidopsis. What it does in a cell, it has multiple organelles. Cell has multiple organelles, right? The RNA is made. The RNA has to go where? The RNA has to go to the plasmid. Why is it? It is the cell. Am I correct? There is a quite amount of journey from nucleus to that cell itself. That is facilitated by cyclophilin. That's the lecture paper in the last year. So you can imagine that what the emphasis is being given in order to understand that what it is and how it is. Okay, so I will try to highlight some of this. Exosome mediated. Exosome was not believable. When I joined IISER Pune, people asked me, and I said, I don't believe it is going by exosome. 20 years later, Matredi, I wish I could have been smart enough to understand that there is something else going on. 20 years later, there is a science paper on exosome. RNA molecules are gathered on exosome. Exosome is by pinching off going to the apoplast, going to the other. Just imagine. This is really very exciting, inspiring, as well as whichever way you can categorize and for our patient. Mind blowing. Yes, right. Now I will give you an example. I must have shared these slides in Bose Institute or some it may be new. I mean, some of them have already seen that. Now the point is, did plant biologists never knew that what causes a plant to flower? What is exactly? If you look at 1987, when I was a student in college, I have read plant physiology book. This has the clearly written the florism causes plant to flower. Now, what is that bloody florism is? Is it a amino acid? It's a metabolite. It's a hormone. Nobody knew. Decades gone. Biologists worked. Decades gone. Now, this is a clever experiment. That's what I was hearing, that how Professor J.C. Sengupta was careful enough to understand what is happening in the jute, why it is early flowering. Okay, now there it is. Look at this. It is, I don't know whether you can see it here. Chalikan, 1981, 40 years back. I think 40 or 45. Okay, whatever. What he did, he took the tobacco plant, flowering tobacco. Okay, he took the tobacco plant and grafted onto potato. Why did he do that? Because he had a hypothesis that this flowering and the tuber development, both are reproductive phenomena. If there's something common, something might happen. With that intention, he grafted tobacco on potato, a plant potato which does not produce any potatoes below ground. Fair enough. Done. He grafted it, left it. After a month or two, when he uprooted this graft, he found the plant has produced tubers. Correct? This plant, it's in front of you. You don't have to see me. It's right here. Plant has produced tuber. The question arises: where did the tuber come from? Did Chalikan was able to explain that? No. But Chalikan's fundamental curiosity-driven one experiment has set the foundation in the last 25 years. Any experiment that biologists are doing, highlighting his experiment, that clicked others to identify signals across the graft. Did you follow my point? Because something must have moved from here. What it means that a flowering signal could entice a potato plant to develop a tuber, suggesting that the flowering signal and the tuber signal could have similarity somewhere. I do not know. You have to explore. but it is happening in front of your eyes. You do not require a molecular Western, extern, Western RT-PCR or Northern to see that. That's what is happening. All right. So this is fluorescence. This is fluorescence 1987, 1990s. Nobody knew. Still, we continue with fluorescence. Textbooks have fluorescence, and the discovery. I also written the answer in my graduate classes. Now, what happened subsequently in 2005? There was a science paper came up. People showed Tao Huang and Ove Nielsen. What they showed that mRNA of the Arabidopsis gene FT, it is also called FLT, flowering blocker T. It moves from the leaf and it goes to the shoot apex and acquire the competence of flowering and plant flowers. A science paper published in 2005. You can imagine 1935 onwards, the fluorescence was going on. 70 years, biologist has no clue. Textbook has fluorescence. 70 years later, science paper came. People were excited all over the world. Wonderful discoveries. mRNA moves from leaf to the shoot apex and allows the plant to flower. That much is enough for you to understand. But you see the trajectory of that scientific discovery. 2007, unfortunately, this paper science has rejected, and the group had to retract that science paper from the Science World's leading journal of science. For what reason? Tao Huang, whom I met, Tao Huang was unable to explain what he did. He was not able to bring enough evidence that mRNA, he was quite close, but the experiment was insufficient to prove that mRNA is indeed moving. mRNA does move. 1995 experiment, I showed him, mRNA moves, but he was unable to claim that. 2007, this paper got attracted. We again got back to the fluorescence. So 2007, by the way, flowering, any plant before it flowers, flowering is a very complicated mechanism. It doesn't go on by one factor. You require multiple pathways, you require multiple factors. Combination, outcome is flowering. There are multiple factors here, which I'm not highlighting today, just telling you, the students, that is called FAC. Floral activation complex must be efficiently produced in the shoot apical meristem. Only there is an outburst of flower, otherwise not. And we have seen it. Okay, so this is flowering. I will come back to this. Now, 2005 I mentioned. 2007 in the Science Journal, in the same issue, back to back, one after another. There is no page gap. One after another, two papers published from two different groups. One showed in rice and one showed in Arabidopsis that FT protein moves. Now imagine 2006 that paper got retracted. 2007 the group came up. This world is, I mean, there are you can imagine the scientists who are working. They say protein moves, mRNA doesn't move. Previous guy was wrong, we are correct. World debated over years. And finally, today we all know that Tao Huang was partially correct. FT mRNA also moves, FT protein also moves. Now, does plant require mRNA and protein both? Answer is yes. You can see the literature. Yes, it is. It is. Because there is an efficient formation of the FAC, the floral activation complex, that only outcomes to the flower. mRNA and protein both moves. 2007, both paper in the same journal, same issue, back to back, two paper published, showed that FT mRNA move in rice. It's a FT homologue called heading date 3 in rice, and it is an FT in the Arabidopsis. Wonderful discovery. Now, I mentioned to you that the activation of signal right here, the activation of signal has to go here. It has to interact with multiple factors. Finally, there is a competence of the floral competence, and then only there is a flower. Ultimately, you see the flower. All plants do not flower. We don't understand why they don't flower. Well, I'm not considering here the photo period. I'm just giving you the general analogy of flowering information. Now, I work on potato and how I connect this information to my ongoing science. 2007, please remember this year when two science papers got published, and we were working that time in relation with Tao Huang's discovery, which got retracted. What is happening in potato? I'll come to that. Just have these questions in your mind. Now, not only 2007. Subsequently, what happened? Many other groups jumped into the field. They realized the potential of this understanding of the signaling mechanism through the mobile RNAs and proteins. This is another classic example. Yesterday, I discussed in IISER Pune, IISER Kolkata, and I showed that Hi5 is a transcription factor which coordinates light and temperature. And if it doesn't coordinate, if you mutate Hi5, the plant's growth, development, physiology is completely affected. Hi5 is a crucial factor. Now we know today, and it's a current biology paper in 2016, they showed that Hi5 moves from here to here. And what it does, it establishes a balance of carbon and nitrogen requirement in the plant. You mutate high 5, the plant's carbon and nitrogen balances. And they have clearly demonstrated that it is moving from here to the here. Okay, that's the discoveries. To move on this. Now, this is another important questions in front of all of you. I have written seed maturations and fruit ripening. In my opinion, it's a conundrum. I say it's conundrum because why I say it's conundrum? It's a difficult question to understand. On what basis I claim this? Because the seed maturations and fruit ripening. A fruit may kick off its ripening process. Seed maturation, seed may not mature. Fruit may be ripened, but seed may not mature. Or seed may be mature, fruit may not be ripened. This process is tightly integrated. And the papers today, the leading groups in the world have demonstrated carefully that if this process is not efficiently managed, you will find a fruit while the seeds are not mature, the ripening process has kicked in. And I'll show you one example here. I'll skip the video, by the way, but you see in case of apple, the Malus domestica, the apple, you have the right-hand side, you have the ripening process, you have the unripened. Hundreds of mRNAs have to move from this stage to the other side. If it doesn't, the ripening process is affected. You cannot. It's happening in tomato. It's happening in apple. It's happening in fruit trees. It's very clear to us. Now, you need to find out crop by crop, of course, but there are demonstrations in particular fruits and particular crop plants. Suggesting that, what it suggests you that these mobile RNAs and mobile proteins have tremendous impact on the growth and development. They are. Now, you can say, did plant produce these mRNAs before? No. During the developmental stage, those mRNAs are required to move. That's why plant produced them. Okay? Because you don't find them before. You go and do any scanning, any sort of laser. We have done laser ablations, many things we have done. We have gone cell by cell to see, was it produced before? Certainly not. They were not. This is, of course, the example of SEC oxidase. SEC oxidase is the gene which is the key enzyme in the ethylene biosynthesis, the last enzyme which you perturb. If you make a mutation of ethylene biosynthesis, your ripening will be affected. Plants will have not the ripened fruits. Clear example. And ethylene biosynthesis, by the way, is a demonstration of this paper in Molecular 2024. One shows that it's a mobile signal. Now, is it only what I did? Again, I said that because I took an opportunity, this endowment opportunity, to let you know that the field, I mentioned that it has an impact on the development. Mobile RNAs are controlling the plants. I mentioned it has an indeterminate growth. There is a reservoir of stem cell. This stem cell, there's a washout and there's a clavata. Then it requires to interact and make a feedback loop to maintain the stem cells and keep the plants always growing. That's tough job for a plant. Now, there is I don't know how many of you know, there is a L1, L2, and L3. There are specific layers of cells. The upper layer, the second layer, the third layer. Cell layer positional information is very critical for a competence to achieve, and the growth has to happen in a real-time manner. Now, these positional informations are achieved by careful targeting of, careful export of mRNA and proteins from a below cell to the next cell. That's one of the examples in front of you. The shoot meristemless is a fantastic example. This is, of course, by tagging and you have YFP, GFP, and you can see that where is the RNA has gone in terms of Arabidopsis. If you look at Arabidopsis and do the developmental stages, right from the zygote to you just track it from zygote to the next stage, to the hard stage, to the cotyledon stage, to the next stage, you will find that the mRNA has moved. It was originally not there. It was synthesized and moved. This is one of the examples which does that. So this is a description of here. STM mobility is critical for its normal function in stem cell control. Otherwise, stem cell will not be controlled. Plants will stop growing. No, impossible. STM controls CAC expression. So you don't need to remember CAC, what it is, a transcription factor. Post-embryonically, after the zygote formation, at organ boundary domains, boundary must be mentioned. Otherwise, leaf will develop in place of shoot, shoot will develop in place of leaf, sepal will develop in place of petal, petal will develop in place of sepal. You require a correct boundary. Imagine these fine-tuned boundaries are maintained by positional recruitment of your signals in that particularly. The signals are not moving to the next. They are there. They are there to what? They are there to repress the transcription factor. They are there to activate the other genes in order to function, to achieve in a real. Making sense? Just raise your hand if you are not. This is another example here, right here. You have cucumber and pumpkin. Now take this example. This is from Bill's lab. So you have a pumpkin stalk here. This is a cucumber here. The cucumber is defective in growth. It doesn't grow very well. Now they grafted cucumber and pumpkin. Now what happened from pumpkin? You see a beautiful transport of a signal from here to this here and allowing this guy to grow. And that's what it is. Fluent transport of NAC. It's a NAC domain transcription factor necessary for plant development. Plant growth and developments are controlled by multiple transcription factor. NAC is one of them. Was proved directly by heterograft. It's a heterograph. Homograft and heterograft I did not mention. Homo means same to same and hetero means two different classes. It's a pumpkin and cucurbits. And transcripts are shown to accumulate in the cucumber cyan right here. The transcript of NAC has gone there. It's either a antisense or a silence lines of NAC. Thereby the transcript, if it goes from here to here, you can carefully identify the transcript has moved. And the plan shows, of course, physiology of development. As I said, you do not require a Western to prove that. Okay, so I just mentioned abrasionesis. Those of you are familiar. At the early stages of plant development, the growth is restricted. And this specific small RNA, it does the amazing job. It just line up here. It gets expressed at the line of development. And it cleaves another transcription factor. It's called leaf curling responsiveness. Thereby this growth is achieved. And finally they go to the trajectories of them. How do you challenge this kind of experiments? You go and pull out this paper from developmental cell. They have done the microRNA mimicry, microRNA sponging. They blocked the microRNA. Now, the moment they blocked, the leaf curling responsiveness expressed very heavy amount. The overall development got perturbed. It is showing now stark developmental effects. Right? That is what it is. We are sharing with you. This is another example for the leaf. I'm giving multiple examples to you to convince that yes, it has an impact on development and growth and development. Here you see you have a mutant graft. It's a stalk. It's a cyan. Now you look at the wild type leaf, tomato leaf, how the leaf is and how does SEM look like. The mutant leaf, look at how the phenotype is, more of a compounding, and look at its shape and look at the SEM picture. Now the wild type is grafted on mutant, and the mutant, it's a leaf signal, right? So its signal has moved from here to here. It has changed the wild type leaf completely. Look at this. Clear demonstrations. So there is a beautiful transport of signals from the stalk to the one, suggesting these signals are essential for leaf development. They are leaf signals, leaf cell development signals. There are plenty of literatures, but I just picked up for these lectures very carefully, selectively which one I felt very important for us to know, enhance our knowledge, and it was knowledge for me as well. So, and there are LNC RNA. I told you that gene regulation is going to a much deeper understanding. LNC RNA is a now new player. We thought microRNA is the end, but no, now we have LNC RNA, and we don't know how LNC RNA is controlling the gene regulations, giving the developmental different sort of outcome, physiological perspective, how it is controlling the development. So we now know LNC RNA moves from here. In a nutrient stress, in a senescence, ALENA1 reduces the senescence. Now, if you do the grafting, it shows you that ALENA1 moves from the root to the leaf and controls the senescence of the plant. That's the example is published in Nature Plant 2 years before. These discoveries are amazing discoveries in the field. Okay, so I mentioned to you that plants can make multiple mRNAs, but not necessarily. We do not know which one is the mobile signal. All we know, there are proteins which bind to mRNA, make a complex, and then it goes from one cell to another. Now, this recent discovery is a Nature Plant discovery. Again, what it has done, they have identified that in the cell there are multiple organelles. There are vacuoles, there are Golgi modules, there are mitochondria, there are other things. There are organelle specific proteins. They are binding to the RNA and allowing the RNA to go and reach to the cell wall. But where it has to go and line up with the plasmodesmata to go out. Because in the plasmodesmata, there are other proteins which will bind to it and take it away. Okay, that's the discoveries of this. And this mechanism is now clearly known. Plasmodesmata plays important role. Its gate, its aperture, you can imagine. You can maybe thinking about, does everything go? Does it has a limit? Whether it has a restriction? It does have a restriction. it does have certain control of its own. But we have seen case by case, there are evidence by evidence that what we can now see that some of the proteins can bind to the plasmodesmata associated proteins in that collar, in that sleeve, in that desmotable, and can modulate plasmodesmata shape and signal can selectively, again selectively, nothing that it's a default mechanism. This is one of them. It's a caprice. If you make mutate root hair, so important organ for a plant to absorb nutrients. If you mutate caprice, there is no root hair. Oh, wonderful. Now, so what caprice does? It's a mutant in front of you. See that the mutant has no root hair, and this caprice, if you tag with 2x GFP, 3x GFP, 4x GFP, 5x GFP, what we see that it can dilate, not dilate, I would not use the dilate word, it can modulate the plasmodesmata aperture and can pass the signal, thereby controlling the root hair wherever the root hair is not developed. So caprice is the signal for the root hair that we know today. I mentioned environmental factors play a critical role. All the environmental factors that I mentioned, light, temperature, biotic, abiotic. Our lab, we are working on potato so many years. Potato, unfortunately, is very susceptible for a disease called late blight, all of you know, and Phytophthora hijacks potato systems. So I said, I don't know, I'm not a pathologist that I would study, but all I would like to study that whether there's a signal being generated at the source and the signal gets transmitted. That's what I wanted to know. One of my students was very interested. I said, yes, if you're interested, do not understand the pathology. We are not expert, but we can understand, we can pick up the signal. And what she did, one cartoon can help you to know what she did is that, so it's an infection right here. This infection quickly generated signals which were all present on the distant side. This signal is nothing but a small RNA signal. It's a microRNA 160. First time we showed. This paper, I mean, a lot of reviewers, things happen. We always think the paper should go even more higher journal, but it's a tussle for all of us to continue. So that's what we showed, that yes, signal I can again show you, signal can be generated here, and the signal can ultimately go. It goes by the phloem. Catching a small RNA in a phloem is not a trivial task. You can imagine how challenging it is to identify that is indeed a signal or not. So this is what we showed so far. Here is an example in front of you. That this is what we common example in our all the time that we notice, but hardly we pay any attention to it. But you imagine the amazing discoveries are coming up. This guy is hijacking the host. It's hijacking, and how does it hijack? It doesn't have any leaf, doesn't have any root, doesn't have any stem. It's not a true stem either, but Cuscuta grows happily all its life. Most importantly, you take a piece of Cuscuta and put it somewhere, it again grows. The development we don't understand. I'm sure there will be a science or nature paper someday. But in terms of its hijacking systems, I'll show you a slide and you see what it does. All it does is that it wraps up the host and it generates hostoria. Hostoria is a kind of a structure. You see here, it's the host, and this is a Cuscuta, and Cuscuta is developing some hostoria. Hostoria is piercing the host and getting and touching the vascular bundle of the phloem. Why phloem? Why xylem? It does xylem and phloem. It crosses xylem. See, there are external phloem, there are internal phloem. It crosses xylem. So basically, from xylem, it hijacks the sugar and things like that, and other things, the nutrients, whatever it is. And phloem, it goes and what it does, it recruits its Cuscuta signal in tomato. Now, why Cuscuta is putting its signal in tomato? It's simply to make the plant more susceptible, binding its receptors, binding its protein, so that that guy goes under its control. On the other hand, the host, what it does, it also transmits its signal to the Cuscuta. This mechanism is done by exosome mediated. I have given here multivesicular bodies. You have the exosome. Exosome is released in the apoplast, which is now all the plant mRNA. It goes and blocks the defense, and thereby the mechanism is involved. Okay, so I hope you are able to figure out that what is happening in the field of this kind of mobile signals and its effect in the plant developments and growth and productivity. I work on potato. That's my lovely crop. Been working for 25 years. Like to work for more years. Honestly, anything that I understand, I think I didn't understand anything. But Over the years, we have identified half a dozen mobile signals from this plant. Now, I'll go back and connect you with one discovery that I told you. That 2007, there were 2 science papers. 2007, people have shown that FT in rice and FT in Arabidopsis are flowering signals. Now, in potato, this development is nothing but a reproductive process, like flowering. So, honestly, the hypothesis could be, if the flowering is controlled by FAC, this process could be controlled by TAC. TAC is tuberation activation complex. Flowering is flowering activation complex. You require not only one factors. You require multiple dynamics of hormones to play here to bring this outcome for us. It's not an easy. On the other hand, you require multiple factors. You require temperature. You require light. How is it easy to understand? Simple thing we asked: what is this signal? The questions we asked in 2001 when I was in US, and 2006 when the field is very hot. People are debating over the mRNA versus protein. We came up with a signal. We said in potato, there is no protein signal. It's mRNA signal. And we sent out the paper to finally, we were successful in demonstrating that in potato, I'm telling you in a summary, that there's a signal which is made here. And the signal goes all the way here. Misdirection of that signal can produce a potato anywhere in the plant. And we have shown that. So you need to believe. You do not require, again and again I said, you do not require my RT-PCR, do not require my Western. You require the phenotypes, which is what is the clear outcome of the result that we believe, or the hypothesis, the question. I'll show you that. I'll show you how we did that. So this is our process. Please remember one more, please take a note. I should not say remember. Please take a note of one more important point, is that this process is, I've already mentioned, there are multiple things control this process of development. It is not only some hormones. In the phytohormones, there are 6-7 phytohormones are playing role. How much we know, how much we have understood so far. That's our most our interesting and investing our process of mechanical. I highlight here the organ that Moitridi mentioned is a stem. It develops below ground. Potato is nothing but the stem. It grows below ground. Now you may ask yourself a question, that being a stem, why would it grow below ground? First of all, you can come up with your own answer. If you don't, you can ask me. So the point here is this stem grows below ground. Now it doesn't grow in the beginning of the juvenile phase. It grows when the plant is going to the adult phase. It's a reproductive fate. It's a reproductive outcome for the plant. So now this organ which goes below here, remember this organ, it has a dual fate. What do I mean by dual fate? This organ, by some means, if it perceives the light, there is no tuber formations in the plant. But you need to know why, number one. And if it does not perceive light, you will find plenty of tubers. Farmers are happy. Okay, that's why farmers cover the plants with more and more soil as the plant goes towards the maturity. It covers. They spend a lot of amount of time in the field to cover the base, to protect the organ, which is the time it will develop numerous organs. They would expect all of them to turn out into a tuber, but some of them again go and then. Now this is where I'm coming to the fact. In potato today, I'm claiming we have identified multiple mobile RNAs. Are all mobile RNAs are towards for the tuber development? No. Some are acting as a positive regulator. They are inducing the tuber development. Some are acting as a negative regulator. They are repressing the tuber development. But why you think activation repression mechanism is essential for a plant to establish when they develop an organ? It is essential. We have shown that. You need a repressor. You need an activator. Why do you need a repressor? You please take a note of this. This tiny head is nothing but, which I have shown you examples, is nothing but a SAM, the shoot apical meristem. This SAM, there are stem cells, there are programs built, it can go to a plant. You require all the STM, you require OCL, you require clavata, you require all the SAM markers to be those of you are taking photographs, please do not share, to be established here. On the other hand, this organ also has to accumulate what? This organ has to accumulate sugar. Where is the sugar coming from? Sugar is coming from right here. Now, in synchronization of sugar delivery from the top to the below ground, this organ, when it grows, it has to change its developmental outcome. What I mean, the cell division pattern must change. It from radial, from two different cell division pattern, it has to control right away. It has to swell up. This is the organ. You can imagine my folded hand as an organ. If it is the case, it has to swell up and integrate the upcoming signal coming from the photosynthetic product has to go and accumulate in that. That's how the potato is. And there, what is necessary for you? You necessary in one hand, the positional information deducted by those mobile signals. Your potato signal has to come in, your tuberation activation complex has to happen, your hormonal control has to happen. You have 6 hormones, I did not mention it. Gibberellin must go down, otherwise plants will not produce. So you need multiple transcription factors, multiple signals, which has to all come from the top and control the tuber activation complex. That's the outcome of right here. Similar to flowering in the potato community, people thought it very easy. They published paper. So in potato, it's another tuber region. Wonderful. Paper got published. Tuber region. Tuber region did not. People 2 decades gone. We did not know what tuber region is. Now in 2006, what we showed in 2006 that it is a mRNA which is moving from the top and inducing the battery of tuber development. And you come to the slide. This is the dynamics that we are interested to understand what is happening here and how this organ is finally formed. This takes roughly about 25 days under inductive conditions. You require inductive conditions means light. Remember, he was saying that Professor Sengupta's observations in jute, early flowering, and the quality compromise in the jute. Similarly, there is the photoperiod which plays a critical role here as well. I mentioned to you hormonal role. Gibberellin and auxin plays important. They have an antagonistic relationship that we know now. Gibberellin must go down, then only plant will produce tubers. Otherwise, plant doesn't. Not only that, I also mentioned to you there is a complete different radial to radial cell division to what is opposite of radial? The other longitudinal. Exactly. Thank you, Majidi. So radial to longitudinal cell division change pattern. This change requires to establish by how? You require those signals to come and maintain and give the direction to the cell that stop radial expansion, because your sugar is now inflowing. Sugar unloading, sugar offloading is another conundrum for us. We do not know. Right to the cell. And that's how the signals are playing out here. What we did, we identified a bunch of transcription factors which are necessary for growth and development. I do not want to go to all. I will show you one highlight here. We have discovered this BEL5 is a transcription factor. mRNA of this protein is a mobile signal. It's a 2006 discovery. Was it shown somewhere? This BEL5 interacts with many other NOX in transcription factor. This interaction is super important for plants' growth and development. Take a note of that. Not only this interaction of these two, what it does, BEL5 binds to H1, it's a NOX1, makes a heterodimer. This heterodimer goes and binds to the gibberellin biosynthetic gene and repress the gibberellin synthesis. Plant must kick in to the tuberation step. Right? That's how it works in the physiology context. If you misdirect this signal from leaf to the stolon, this is a clear example. By the way, we showed this BEL5, the first mobile RNA discovered in potato. When we showed in 2006. Imagine when the science paper got retracted, we came up with analogy that in potato it is nothing but a RNA. I got numerous mails. I said, you're most welcome to challenge. We will show you that it's mRNA as a signal. Subsequently, what we showed that if you misdirect that signal of BEL5, it produces tuber other places. So that's a clear example. How did we do that? Students may be thinking that what sort of experiments they did. We did a simple northern experiment to see how the BEL5. Our hypothesis was BEL5 is likely a signal here. Either it's a protein or mobile. I do not know. But we did a clear northern. By a northern analysis, we could find out that 2 different photoperiod trajectories. Your accumulation of RNA is different. And that gave us a clue that maybe it is something going here. You can see here you have a long day and a short day photoperiod. Plants is exposed to different light, and we did a northern analysis. But under short day, you have an RNA which is accumulating more than 10-fold compared to long day. Why would RNA molecule, immediately you can ask a question, why would RNA molecule will be accumulating here so much? Now, does it mean this RNA molecule is repressed from translation? Indeed, we showed that RNA is way more stable. 90 hours, this RNA doesn't degrade. We looked at 15 RNA molecule. to prove the community that look, all others are degrading within 15 minutes, 30 minits, but this guy remains pretty stable. Now, does it make a protein? I don't know. I don't have antibody either. But this guy, this BEL5, it moves from here and it goes here, and it binds to NOx. I showed you that it binds to NOx. That we showed that. Indirectly we showed it. Goes and binds NOx there. Why it binds there NOx? Because it has to bind and control the gibberellin biosynthetic gene. Plants have to reduce the gibberellin to go to the reproductive rate. That's what we showed. How did we do that? I followed Chalikan. Chalikan is the magician, I would say. I followed his work. I said, let's go and do a graft. We did a grafting here, and this grafting resulted amazing observation for us. We grafted here, you see that? I grafted this guy here, and finally, this is my cartoon, of course. It's a very old slide. I prepared more than 20 years, almost 2006. And ultimately, you have tubers here. Okay. Now, if you have tubers here, so that means something has moved from here, right? Something has moved. Obviously, we'll expect something has moved. Now, what is that something? By doing careful laser capture microdissection, by doing phloem-specific transcription assays, by hooking BEL5 to phloem-specific promoter, we were able to show that BEL5 expresses in a very different manner under inductive conditions versus long. Now, if you do not do that, there is a misdirection of BEL5. The tubers are not produced below ground. Fair enough. Okay. Not only that, if it moves, it makes more tubers. It makes more tubers compared to, that's the result here. It makes more tubers. It has the largest UTR sequence that compared to all other BELs and NOx that we knew. It has that. Now, I was challenged. I said that if your BEL5 is a mobile signal, and you are claiming that your UTR is the key where likely the protein discoveries are much later. We have done like several years back. Earlier, we did not know that proteins are binding to the mRNA. But we found out that BEL5 is the largest UTR, and UTR is loaded with polyUs. So there must be proteins are binding, which we identified much later. Now, in between, I was challenged, and I said that if you are claiming that your hypothesis is true, then ideally any junk RNA should be able to move. I said, yes, why not? Let's try and see. So we took a junk RNA, we removed this BEL5 home framer, and we put a junk RNA, which has nothing to do with the mobility, and we made a construct and we put back to the plant. The junk RNA got delivered to the below ground. Now, how did I prove that? Because I removed, in some cases, I did a control, I did many controls, negative control, positive controls, and thereby I showed that your junk RNA is moved, and finally we were able to publish and show the community that interesting observation. BEL5 is a mobile RNA. It induces tuberations, it requests. BEL5 is the first mobile RNA in potato. Is it all? No. We identified there are RNA molecules which are acting as a repressor. BEL5 is a positive regulator, but there are RNA molecules which are repressor. Why are you doing a repressor? I showed you that mechanism. I have a movie. I think that will help all of you that what is happening there in the below ground. Let's skip this. Doesn't matter. We identified RNA binding proteins. These proteins have RRM. RRMs are RNA recognition motifs. It has 4 RRMs. We have 6 PTBs, the polypropylene tract binding proteins in potato. We identified them. We did a careful analogy. We did one by one analysis, and we see that 2 out of 6 PTBs, the binding proteins, are positive regulated. They're inducing tuberage. Now they're inducing, BEL5 is inducing, is very, very, very chaos and chaotic situation. We do not know what is happening. Finally, we came down. We did a lot of RNA EMSA and things like that, and thereby we identified that this PTB is the bind to the BEL5, and then finally goes to the targets. Subsequently, we have shown all that. This is how it is. When a protein binds, you can see that that's how it looked like. It's of course a reference from Q Curvit, from Bill Lucas from University of California. That paper got to plant cell, ours is going to plant fish. What we showed here that your protein binds to the RNA, and then the RNA got delivered to the target site. This is how it is. It has nothing to do with the gravity. It has an independent of mechanism. It can go to any directions depending on the need, depending on the target site, depending on the strong sink or weak sink. Okay. So I'll come to this and to help students understand. So BEL5 is not subsequently many other groups like that we now know FT protein and mRNA both are signal full flowering. People have identified proteins as well. And this is one of the examples, SP6. SP6 is from Salome Pratt from Netherlands. She has done that discovery. That's a Nature paper. They showed that SP6 is synthesized here. The protein has to go below. Only then it produces the tubers. Now, Do you require protein sp6? Do you require BEL5? Do you require repressors? Answer is yes, certainly. And we have analogy to help others understand that it is necessary. So if you see these all the signals, there are NOx, there are NOx transcription factor, there are BEL transcription factors, there are mobile RNAs. In this, we have also added microRNAs now. It is making our job way more complex. We have now identified 2 microRNAs, which are also mobile signal. I will show you that. So these are made, and these are all delivered here. And subsequently, repressors, proteins, all are, I'm just getting students understand that how it is. And finally, you have the tuber. That's the outcome. Now, this is the structure which grows below ground, right? I showed you that. This structure, what is this structure? I think students should recognize. This is nothing but a SAM. This SAM, developmental, but it's very interesting. So this SAM, it will not grow as a plant. There is a SAM built there, and you require SAM markers to be there. It is all there. We have seen that. Now the point is, this one, you see this, there are repressor molecules which will make it in that state. Cell divisions, radial to longitudinal, will not change until those molecules are coming in here. You see this. Just 1 second. Finally, you have the tuber. The repressors is not making the plant to make tubers, but you have a positive regulator. It is changing the divisions and making the plants to make the tubes. Okay. So this is again similar to flowering. There is a TAC, the tuber activation complex. Not only one protein. We have 14-3-3. We have FD. We have FT. I mean, it's a multiple things that we now understand, and we are still working on this field. Okay. So how is the tuber made? Tubers, the signals are generated here. There are activators in the signal. The signals BEL5, sp6, they all move from here, from petiole, and finally they come to the stolon, and ultimately you have the tuber activation complex, and thereby the tubers are formed. Okay, STBEL5 is known as a transcription factor that works in tandem with NOx type. Both transcription factors are necessary, must interact. We have published papers. We have shown that it is essential to interact in order to balance the hormonal homeostasis in the cell. Otherwise, plants will not produce tubers. Fundamental mechanism, yes. Okay. Now, the question is, I said to you that we have identified many signals. This is one signal that we have identified, microRNA 156. One of my students was very interested. She's from microbiology, and she said, I would like to understand your concept of RNA transport. I said, you're most welcome. So when she joined, she said that I didn't work on plants. I said, you don't need to work on plants. As long as you have the fascinations and the passion, you can work. The point here is, she just simply followed my experiment. She said, let's look at how the microRNA 156. So why, you may be thinking, why she picked up microRNA 156? There was a Cell paper published in 2009. In that Cell paper, people showed that juvenile to adult phase is being tightly controlled by 2 beautiful regulators. One is microRNA 156 and 172. It just antagonistic balance makes a plant to start juvenile to adult. It can retain in juvenile period if you control the 56. People have shown that. It can go quickly to the reproductive phase if you tweak the 170. So the advantage of the crop breeding, advantage of the agricultural point of understanding the fundamental regulators are tremendous. So what she did, she expressed microRNA 156 in potato, constitutive expression, and this constitutive expression resulted. It's very clear in my laptop, but I hope you can make it. It resulted aerial tubers all over the plant. We were surprised by this striking phenotype because it was beyond my expectation that plants will produce tubers above the ground. Then Sneha said, let's experiment it. So subsequently, we did a lot of experiments to understand that why plant is producing aerial tubers. MicroRNA, not only that, we have also established microRNA 156 is a mobile signal in potato. Through grafting, we have shown that microRNA 156 requires to move below ground, otherwise plants doesn't do that. We have done that. Okay, we'll keep this. Okay, so this is a summary that how many mobile signals are now known in potato. All are necessary. Some are necessary for shoot development, some are necessary for repressor, some are necessary for positive regulator, some are necessary for cell division control, some are necessary to control the hormonal dynamics. These are all signals because it's an activation center for me in the below ground. Okay. I will show you these slides and explain to you because we wanted to understand what it is. There are mobile signals, but fine, I know I can make sense of it. Why mobile signals required to develop. Now, subsequently, what we show that this kind of developmental outcome is something different. Something different. Different means you go and look at those. If you simply go and scan this area, look at this slide. If you go and scan this area, we came up with thousands and thousands of genes that are all through a RNA-seq, and that RNA sequencing gave us a different sort of a direction that this mechanism is governed by epigenetic mechanism. How I said it? Because we have looked at those genes and found that all of these genes have either activation or repression mark. Why do they have activation or repression mark? Because they are controlling what? They are controlling more than 10,000 genes which are necessary for tuberations by either activating them in a spatiotemporal manner or repressing them in a different developmental outcome from the plant sector. This is an epigenetics clearly, and we have also, of course, in 2021, we have shown that these are marks. By the way, those of you know how the activations and repression marks in the genes in the above ground and the below ground, the genes we have. Mechanism of tuberation is complex. I've been saying it quite a few times, and you see the regulators, the pathways, what are any mechanism is of course complex. One has to delve in to understand how it is. And again, the plants are, I showed you, it responses to inductive photoperiod. So what happens in a non-inductive photoperiod? Which are the regulators? Which are controlling that? All sorts of questions. This is how all this, what are red marked here, you can see under short day photoperiod. These are all expressed in the above ground. They need to go below here, and then activation of this, all the protein 1433 and other protein, and thereby the plants are producing this. On the other hand, you have, this is auxin and CK levels has to go down, and on the long day you have RAP1 binds. So you can think about why long day plants don't produce tumors, why short day is only favorable for producing tumors. Well, what we have seen in long day, you have a RAP1 which is highly elevated. RAP1 goes and binds to BEL5, remains BEL5 repressed. BEL5 cannot move. It's a long day, non-inductive, non-favorable condition. RAP1 goes high. In the short day, RAP1 is low. Your BEL5 is high. BEL5 is moving, making beautiful analogy and understanding that it is how the tuberation works. Whatever I said so far are the fundamental information, right? The mobile signals which are impacting the plant growth and development. Now, if that's so, we have identified, our lab have identified multiple mobile signals in potato. We have given analogies. We are asking people to let us know that is the analogy making sense. Now the question is, we have identified RNA binding proteins which are acting as a positive regulator. We have identified mobile RNAs. One of my students came up and said that you have all these signals sitting in the lab. What are you going to do? I said, I don't know. He said, why don't you make a gene pyramiding approach? I said, fine, let's do a gene pyramiding. What does gene pyramiding mean? You take those mobile RNAs and proteins, and you look at you can just look at this construct. We made a construct, okay? So construct means if my fundamental information is true, it should show the reflections in the plants, right? I claim that we have identified proteins which are positive signals. We have claimed the mobile RNAs are positive signals. Now we took those BEL5 RNA and the two proteins together and expressed them under their native promoter, and that's the construct you see here. It's quite a big construct, almost 24 kb. It was quite difficult for us to move into the plants, but we were successful in potato, and we generated large number of plants, hundreds and hundreds of plants are generated to see what is the outcome of that signal. Because what we did fundamentally in a cell, now I have bumped up the mobile RNA, I have bumped up the protein, I have bumped up the two proteins, and I said, let's look at either the plants will collapse or it will show a abnormal phenotype, or it will not make any tube, or something will happen. Let us be prepared for that and go for it. And to our surprise, when we looked at this, what happened? Amazingly, you see that the number of tuber, not only tuber weight, the number of tubers gone several fold high in that construct, where you have mobile signals. What I did, I carefully targeted, elevated the mobile signals in the cells, and I purposefully wanted mobile signals and proteins to go and reach to the stolon and see whether I can increase the tuberation. And this concept worked. This concept really worked. You have many more tubers. This work, of course, we have done in collaborations with. I'm grateful to CPRI Shimla for their generous help to me, and they have grown my plants into their containment facilities. They have done the tuber analysis. I said it will work. Please go ahead and help us. So they did that. They're very helpful to us. Is that all? We think that this concept, as I mentioned to you, It has a tremendous impact, not only in development, it has an impact on crop breeding. It has why I say crop breeding, you can make early flowering, you can make late flowering, you can make juvenile period extended, you can make reproductive phase early, giving you scope to do fantastic crop breeding. And that's what people are now doing. I mentioned to you, did I mention, I don't remember if I mentioned that the floration is now a big, it's like a big gift to us, because what you can do, you take floration and you do it, take a viral vector, could be apple latent spherical virus, ALSV, or any other viral vector, disarm it, make its virulence go, disarm it, chop the virulence part. Now put the FT in this. Now take that viral vector and just go and infiltrate on the plant. I don't know whether I've shown that signal. I suddenly remember that what happens, because viral protein I have not disturbed. Viral protein moves, movement protein, and FT is now expressed in the plant in early on. Plants go for flowering. So that is another. I think if I'm correct, it's probably a current opinion people have written, that how it is allowing crop breeders to carefully go for those crops which are difficult to cross, having the flower early too, because pollen grains, I mentioned this, I think, and the pollen grains have the capacities. It has a called Xenia effect. If you brush pollen grains and you see the pollen grains effect on the maternal tissues, amazing. Okay, so that is the scope, that is the impact, it is the productivity I was talking about, giving examples from my work. In the first half of my lectures, giving examples from other work, I tried to establish the 30 years, including the mechanism of the RNA proteins, the signal deliveries. I showed you that how many mechanisms people now know, and that how the RNAs and proteins are now moving. And they are not only moving cell to cell, they're also moving long distance. With that, I'll close, and I hope I have given you a glimpse of work and understanding the biology of this field. I must have taken longer than I was told. I'm sorry for that, but this is our institute, Ministry of Science, Education and Research, our main gate. We welcome all of you to visit our institute, and this is our IISER Pune. We have, many of you have not visited, we have expanded 2 more floors here recently, so that's the old picture for me. This is my group at IISER Pune. Generous funding from Ministry of Expenditure, Department of Biotechnology, Department of Science and Technology, SCRB has been very generous. I've been successful in securing money that I need, because it's all our goal. And I acknowledge at the end, again I reiterate, and again I said I'm grateful for giving me this opportunity, all others, for giving me this opportunity to come here, share our work, our passion, what we like to do, what we like to understand. I encourage, I seek your inputs, your difficult challenges, questions that I'm unable to answer, that will help us to move much better. Thank you so much. If there are questions, I'll be happy to answer. If they allow me to answer, I hope it was. Thank you. Hope it was.
What a mesmerizing talk. I believe number of questions are bubbling in your mind, but due to shortage of time, only 2-3 questions
will be allowed. Please go ahead, whoever has the question.
Please.
I don't think I will be able to answer your questions, by the way. Please go ahead, please. Thank you. Yes. No, so no. So Majidi, maybe I may not have put it rightly. You must have been careful in observations. Very right. What is the potatoes? There are they neutral. They don't respond to light. Number one, and there are response to the either for long day or short. The long day, if you put it, it is not going to make any potatoes. Oh, acha. So it's a accumulation of all. So it's what is happening. So the greenness, if you see, This is the stem. Right. At one point, it will start generating its chlorophyll. So you would eventually yes, it is. So I'm saying if you leave it in a it will start winning. What is happening because stem is differentiation is happening. Cell is converting to its original what it was meant for. No, completely de novo induction of. Yes, exactly. De-differentiation. Yes, sir. Yeah, there are questions. Some
energy left for this old man to give some answer. At the outset, we talked about source and sink. Source and sink.
the source and sink, yes, correct.
The single molecule goes from source
Okay.
to sink.
Correct.
If there is no sink,
There is no sink. One. Why would they go? Right. So why would you think there would be no sink? I would say transient sink, not truncated sink. A sink which may be required for a classroom. So very good question. So this question is why would it go the signals? In a developmental paradigm, if you look at a plant when it starts from its germination, right from the germination, energy source, where it comes from, how it grows, how it grows. Then I mentioned one statement very carefully. The source and sink relationship is likely to vary at a different stage of the. A sink may not necessarily be a strong sink at all the time because a growing shoot apical meristem is a strong sink for it. Not necessarily. No, I want in other perspective. So here, so here how it is happening. In the sink, your source is the leaf. We are imagining that the transcription of all the genes and things proteins are happening, the translation is happening in the leaf, and they are going. Like, for example, a leaf here, and the leaf on the other side. This leaf can supply this leaf as well. Absolutely not. But this relationship may not be all the time. The moment this leaf has finished its job, it may supply to the growing flower and growing. Yeah.
the movement of these simple molecules, what is the change? It may be your microRNA
Yes.
or protein or
Yeah.
multiple transcription factors.
Yeah.
They move to the plasmodesma.
They move to the
Micropore.
100%.
And always to apoplast.
From plasmodesma, then apoplast. No, it can go to the next cell as well. Cell to cell, I say cell to cell.
The electrical signal. Like the animal. Action potential. Now, so that I want to know the velocity of the movement of this signal.
Only very recent in the field it is many people are working on and concentrating on understanding the developmental of the concept. For example, now people are taking RNA and trying to chase it and say at what rate. It is certainly slower than the diffusion. Yes, it is much faster than diffusion. RNA molecule. So how it is moving? We are only RNA. Advanced imaging technique can only let us to do more crucial experiments to understand how. For example, as Banerjee mentioned, I have developed a instrument like my lab only with the purpose to do some critical experiment. It is difficult. Seeing an RNA in a potato leaf is difficult for me, but I can see it in a unilayer leaf, which is a unilayer leaf of instrument. You can see unique. See nothing of that. You can literally go by cell to cell under light imaging. I hope my analogy was acceptable to you. Yes. Yeah, Can one, the first science paper.
sand.
Yeah.
Sand is moving to the cloud right? So
Yeah.
the information system and the
Leaf question as well. Yes.
so if it
So, I mean, these are, I don't think we have any, this sort of analogies experiments have off. N1, discovery 125. If you see last year's science paper, David Jackson last year showed N1 mRNA is cracked by another protein in our lobster and taking it to us. It took for them so long to identify that protein. N1 protein itself moving its RNA, and other proteins are also hijacking the N1. I did not. Yeah. Yeah. I also like to use one word, it's called hitchhiking. Many of us used to do in the college time, taking somebody's leave from here to there and from here to there. It's called hitchhiking. We have now hitchhiking process in the cell. You have a protein which is taking it to the Golgi and then gone. And then you have a protein which is binding to it. And I really mind-blowing experiments have been done by people who have advanced tools and things. Amazing information are coming out. Spatial component, developmental context manner, disease context manner, stress context manner, nutrient stress, water stress, drop stress, temperature stress. You can imagine, I mentioned in one set, it is all contextual. The plant is the slide, it has to deal with this, all the mechanisms. I hope that analogy is acceptable.
yeah, last question. For the expression or non-expression,
that I don't know. All I know that what we have in the lab now, more regulators and more observations. Now, are those observations helping us to make meaningful conclusions? Yet to be seen. So we are doing some, but at least two epigenetic regulators. We have seen that they are mobile. Now, why they are mobile? We don't know. We have not.
this is observation that we see in the field. We talked about air users.
Yes.
So where we see predators nowadays is from years. Last year, there was a lot of these air users that we saw in the field.
Okay, naturally.
Yeah, naturally. So that.
I would love to have a photograph.
Yeah, we have. Wonderful. So I just because these are the seeds that have been distributed. Last year there was a problem.
And you see all aerial busts.
Yeah, some of them were
aerial busts. Yeah. So as what can contribute to such an outcome? You can think about. One could be your environmental patterns. Your temperature or your light would have been.
Sometimes in the same field we had
some and not.
And these are
So
all from each other.
Yeah. So then I would say one answer again. If somebody others accept, it's I think much more than classical genetics. It's epigenetics. Not that we know that, because we now see there are several epigenetic marks in those genes, and they are activating in a different manner and controlling the gene regulations. Sorry? No, tissue culture shows. Yeah. Already propagating. Yeah. Yes. Yes.
So you talked about
microRNA.
So do you have checked any time that microsomes have any role in this transport of these things?
No, that part we have not got. What we have shown in terms of SAR, how it is kept. Yeah. No, we have not. I know, but in that paper we have not done research. Yes. So possibly if we go to that business background, we may be able to relate something about that.
So who actually basically triggers that thing? And
only wish we can make a good paper.
That's a query.
That's a query from our side as well. We don't know. Yeah. Yes, exactly. So if you leave a potato there in the home for a couple of weeks, you see there's a beautiful eye formation. But I think what we also said that like it's nothing but a stem. So developmental competence changed. It was a stem. Now it is acquiring a different sort of developmental. So there were several. So it is now showing the STEM buildup. So you will find all the markers of STEM. Yes. Yes. Thank you.
Because you showed RNA movement over long distance. RNA they have a negative charge.
Yes.
I was wondering, have there any experiment been done with electric field?
Yeah. And if I am not mistaken, you asked me the same questions in SRM University when you were in my. And there also I said we have not done.
Getting old.
No, you are not getting old. We were together in the same group. When I was presenting in the he asked me the same thing, right? I don't have answer right now. Yes. So I have to say sorry. But it's a very good point though. It's very good point. Think about it.
And also just pointing out,
Yeah.
JC Bose first.
Yes.
Using his own microelectrode for electrical signals in
Remember, you asked me the instrument that we used, and I mentioned one letter in the hallway of the SRM University. He was taking my test. My test is qualified test.
Oh.
Each signal at the bottom of the plant and where there is a signal at the top of the plant. What happens? If you say that wave signal is always equal to square signal. So a very difficult question to answer. Many people say this is because in the past there is several proteins that changes the electric movement of the. That's what I was wondering. When you say this everything moves to the micro circular matter, what is the smallest protein? What is the smallest protein? What is the biggest size of the
So, issue to issue, leaf to stem, stem to leaf, leaf to medial, medial to other. Organ to organ, the knowledge in the field is that the plasmid size varies. Not only size varies, the frequency varies. So when size and frequency varies, which are the factors which are contributing to the plasmid? That's what we know. There are only few papers in the field currently, because only recently there is a plasmid but a proteome has come up. Now we can do much more experiments because we now know proteins which are in the associated with the plasmid. Otherwise, it was quite difficult to isolate plasmid and go for the Thank you, everyone.
So on behalf of the West Bengal Academy of Science and Technology, we extend very sincere thanks to Dr. Banerjee for giving such a. Let me tell you that I am the furthest from this area.
Sorry,
oh, mic, right? The YouTube, right? See, I am the person farthest from this topic. I am an electrical power engineer. So I know how this electricity and all those things work. But these mobile signals, when you were telling, the first thing that I remembered is this mobile signal. If you ask me, I can explain probably this one, but I have hardly any clue what you were talking about. And on a lighter side, you were talking about activators, you were talking about repressors. And this particular event was an activator because I was in Delhi and I told Professor Shampada that I have a meeting, probably I won't be able to come, but this program was an activator. So I was on the move in the morning. But then, I am coming from Delhi. And so, the fog in winter in Delhi is the repressor. And so no aircraft moves. And I was supposed to reach here well in time, but as you saw that I came almost at the fag end of the program. So that's the repressor was there. But one thing I want to know is that, which gene gives you that much of energy. So if somebody can work on it and put it in me, I will be grateful. I was amazed to see your super secret. Super secret. So it was really that energy which actually I was seeing, and then the involvement with which he performs. And I am told, I know from other sources, not from you, that from other sources who knows you from your workplace, that he's really, not only just an academician, he manages the whole thing so well. Because all these works, if you think, I was thinking in a different way. It's not just science that is required. It requires a lot of management. As he was telling, experiments doing somewhere in Shimla or Dehradun or somewhere, and then all sorts of things. It requires a lot of management, resource mobilization one way, that the funds have to be connected. And I know that in the Indian bureaucracy, how to utilize that money, what sort of activity you have to do. And after that, you have to do science also. And I think you are the perfect example of what the Larsen Toubro chief has recently said, that you have to work 90 hours a day. I think you do that.
I don't know that. But I work in my institute. I have many administrative responsibilities.
I think you surely do that. And keep on doing, Professor Banerjee, and make us proud with your such wonderful programs. And I think regarding other things like the West Bengal Academy and all that, probably in the morning, sorry, not in the morning, at the beginning, with our Vice President, Professor Amit Ghosh, our Secretary, Professor Moitrey Dasgupta, our very senior person, Professor Laidu Majumdar, they have all talked about it. So I'm not going to bore you with all that. I know you are looking forward to the evening. So with this, once again, with a very sincere thanks, I conclude this session. And thanks to all of you. It's a wonderful participation. Thanks a lot.